the exciting thing about stem cells is this whole paradigm is changing dramatically. And what I'm going to leave you with, I think, is kind of a good news, bad news kind of message tonight. And I'm going to also address at the very end some questions that I think we all need to be carefully considering in terms of where the Lord's at on kind of the modern times, what we're doing scientifically, and what the potential outcomes are that are favorable downstream of this whole area of science and medicine, and also the potential downside of what's happening today in the area of stem cells and medicinal therapies. Let me say up front that I am not a stem cell biologist. This is a very unique and specialized area of medicine. But we at the University of Colorado have recently recruited an outstanding stem cell biologist. And at the end, I'm going to tell you a little bit about what his perspective is, and I have no idea about his faith. So I'm going to have you wait patiently for his view of this whole area till the very end. But I think what I thought I'd do to just kind of set the stage for what we're going to be talking about tonight is really show you a video. And this video is not created by anybody in particular. It's not done from a biblical perspective. It's not done from a punitive perspective, nor from necessarily an overly optimistic perspective. But I think it's kind of an interesting scenario that I think is going to set the stage for my talk to follow. So the video lasts about 10 minutes. And then I have a series of slides to share, which I'm going to really be very informal with. I want you to interrupt, and to the best of my ability in terms of what I know about stem cell biology, I'll do my best to answer you as accurately as I can where the current field is at. This is a very rapidly advancing field. And just this morning, when I get a kind of a scientific link called Health Orbit. And Health Orbit every day gives me kind of cutting-edge science and medicine that's published on a daily basis. And even this morning, there was a kind of a new advance in stem cell biology. And almost every other day or so, we're seeing something new coming up. And again, some of it's kind of scary, and some of it's kind of good. So I hope tonight when you leave here, you're going to be a little bit more educated about this very controversial topic. And I think as we take this to the Lord to ask, kind of his view on this, I think that's really where we want to be as we leave here. Everything about stem cell biology and medicine is not on the negative. So try to keep an open mind as you see what we're going to show tonight. So Tony, let it roll, please.
How they work is not important to me as long as it's something that works.
Ideally, people probably should know because it's something, everybody talks about it.
As often as they grab the headlines, human embryonic stem cells remain an enigma to many Californians. What are these tiny biological entities? And why has stem cell research become such a complex issue? Dr. Arnold Kriegstein is the award-winning director of the Developmental and Stem Cell Biology program at UC San Francisco.
Stem cells are really quite amazing. They are individual cells that one derives from an embryo at a stage when there is no specific organ development. There isn't a nerve cell or brain cell or whatever. The cells there are actually pluripotent. They have the potential to become any of the tissues in the body. And you can actually take those cells and in a culture dish self-renew them, maintain them, and in fact expand their numbers. That then allows you to take that building material and start turning those cells into specific differentiated cell types, like muscle cells for the heart, or bone cells, or nerve cells.
Since researchers first isolated them in 1998, scientists have predicted that future human embryonic stem cell treatments may one day work as a super repair kit for the body, generating healthy tissues to grow replacement organs, or even curing cancer.
The embryonic stem cells are really there at a very early stage in embryonic development, just 3 to 5 days after fertilization. At that stage, the entire embryo consists of 100 or 150 cells that look very much identical to each other, and none of them have become fully differentiated. That's the stage at which embryonic stem cell lines are derived.
Stem cell lines are families of self-regenerating cells grown from a single parent group of stem cells. The most flexible of these, and therefore most useful to researchers, are harvested from donated human embryos, usually destroying the embryo. Stem cell lines can also be cultured from the organs of living adults, but these adult stem cells have limited capabilities.
Their potential has been restricted. So if there's, for instance, a stem cell in the nervous system or in the brain, it's what's called a neural stem cell at that point. It has the ability to make other kinds of cells you find in the brain, but it's not going to make the kind of cells you find in the heart or the liver or the pancreas. So it's been restricted. It's no longer what we call pluripotential. The true pluripotential cells that can make all those different types of cells come from the early embryo, and they're the embryonic stem cells.
Because human embryonic stem cells have the potential to turn into any kind of cell, that's made them the tool of choice for most researchers. But they're not without controversy. Some conservatives have opposed stem cell research, saying that destroying a human embryo is tantamount to destroying human life. Supporters, however, note that the embryos used are tiny clusters of a few undifferentiated cells, smaller than the head of a pin.
The embryo we're talking about is really not a fetus. These embryos don't have organs at the stage we're talking about. There are 100 or 150 cells that are a cluster smaller than the dot at the end of a sentence. They are non-specific cell types. There certainly are no organs or limbs.
Embryonic stem cell research offers both great promise and great peril. So I have decided we must proceed with great care.
In 2001, President Bush decided to ban federal funding of research based on the destruction of new human embryos. Even the thousands of such embryos discarded by fertility clinics every year would be off limits.
Scientists were clamoring to explore embryonic stem cells for some time. But when George Bush took office, his decision was that any embryonic stem cell lines that were then in existence, and this was in August of 2001, could be eligible for NIH funding to study.
I have concluded that we should allow federal funds to be used for research on these existing